Well, good morning, everyone. Thank you for attending today's presentation. My name is Eleanor Pinto-Cacosa. I work at Catalent. I'm the technical project manager there. And today's presentation on how preformulation and formulation development can optimize API properties for better drug design. In this presentation, I just want to go over different types of service offerings that Catalent has and how it can be tailored to small, mid-size, and large pharma companies, and also from the pre-formulation stage all the way to eventually commercial manufacturing capabilities. And in this particular presentation, we're going to talk about services that will be catering to more of the pre-formulation stage in your API development. So what is the cost of a new chemical entity? It can cost up to $1.7 billion to bring a new API to the market. And quite often, not a lot of small companies particularly cannot afford the expense of doing a lot of the initial work, particularly the pre-formulation development work, in supporting the work that is needed to bring a API not only to the preclinical trials and development of the material for preclinical trials, but also in supporting it through the different clinical phases. So this is where Catalent wants to give a nice service offering to a lot of customers who are working in this area to support them in their pre-formulation development. One of the industry challenges in oral drug delivery is that the faster and more effective development of treatments with optimized therapeutic performance results in a much faster throughput in developing an API and getting it through the different preclinical and clinical stages. So this quite often results in understanding the molecule physicochemical properties, knowing how to do preclinical and clinical proof of concept, looking at formulation or prototype solutions that can address PK and PD challenges, and also looking at what can provide API stability and robustness of the formulation. And finally, you want to look at prototypes for your preclinical phases that can optimize delivery technologies and also dosage forms. So what this results is in optimal molecule form. It can also result in optimal formulation for looking at the best prototype that can enhance the oral bioavailability of the compound. It can also look at optimized dosage form and also dosing profiles. So as a challenge for a lot of small and mid-sized pharma companies, internal resource constraints can often lead to missed opportunities. Quite often, as mentioned here, with the different types of challenges that can be addressed, such as more molecules in the discovery stage without enough resources to screen them at a rapid rate. Quite often, a desired solution would be a cost-effective option to overcome the internal resource constraints, allowing an organization to rapidly identify bioavailability solutions for their compound. Another challenge that a company could face is an effective parallel screening requiring a specialized expertise and equipment across multiple technologies that might not always be available internally. A possible solution or a desired solution could be working with experts and getting their guidance in the formulation development that is best suited for the compound, and also looking at technology, expertise, and also equipment across multiple technologies that can solve the complex oral bioavailability challenges of that particular compound. So I'm sure, as many of you already know, one of the biggest challenges for a lot of the small molecules that are coming out into the pipeline is that they are poorly soluble. As highlighted by Ralph Lipp in his article that he published in 2013, about 90% of the compounds that are in pipelines are poorly soluble. So this is a big need that needs to be addressed in the industry, and definitely looking at a much more streamlined way of addressing these needs and addressing different technologies that can resolve these bioavailability needs is a very effective thing in streamlining your initial investment that is required at the R&D phase for preclinical testing. And While there are a lot of technologies out there in the market that have been successful in addressing the oral bioavailability needs for poorly soluble compounds, such as the lipid-based formulations, solid dispersions, particle size reduction, and even salt form selection. These are all technologies that are commonly used and definitely technologies that Catalent does offer to their customers. And we do have a more systematic way of kind of offering that to you in our new service offering, the OptiPharm Solution Suite. And in later presentation, I will go a little bit over what OptiPharm Solution Suite covers. I'm sure a lot of you guys know about the developability classification system. This is derived from Amazon's BCS classification system. If you guys don't understand it, definitely I would love to talk to you guys further about it. With this classification system, it kind of addresses what a small molecule compound or even a biologic compound's need is and what the challenges are. Quite often, a lot of the molecules that are being developed either have poor solubility in the GI tract or they have poor permeability into the GI tract and into the systemic circulation. So compounds that would fall under the BCS or DCS1 category would be a compound that is highly soluble and has high permeability in the GI tract. A compound that would fall within the BCS2 or DCS2 category would be a compound that is poorly soluble and has high permeability. A compound that would fall within the BCS3 category would be a compound that is poorly permeable but has high solubility. And finally, the compound that's the most challenging to address is usually the BCS4 or the DCS4 category, which is a compound that has not only poor solubility but also poor permeability. And these are usually the most challenging to work with and hence called the bricks. And in looking at it further, just to address the BCS2 category, wherein a lot of the compounds do fall in that have poor solubility issues, there are compounds that can be resolved by just improving the dissolution rate, as indicated by the left quarter of that quadrant. And there are also compounds that can be benefited by just improving the solubility of that compound, as addressed by the bottom right corner of that quadrant. And James Butler actually looked at different types of technologies, particle size reduction, solid dispersion, and also lipid formulations to see which of these technologies best addresses and resolves the challenge of improving the bioavailability of that compound. And quite often, as you can see, the ones particle size would be most beneficial when working with compounds that are suffering from by improving their dissolution rate, whereas compounds that are have a poor solubility are best benefited by improving not only by solid dispersion and also by using technologies such as lipid formulations. Well, with OptiPharm Solution Suite, we can help improve your molecule at various different stages. With this type of service offering, we can assess the physicochemical properties of the API. We can do molecule characterization. We can do salt forms benefit selection. We can also do preliminary consultation to see what is the stability and what issues the particular API can face, such as thermal stability, such as aqueous or moisture exposure, whether or not it's hygroscopic. These are all different types of things that can be addressed at the assess phase of the OptiPharm Solution Suite service offering. With the next phase, we would look at enhancing the issues that would be addressed at the assess phase by the various different types of technologies mentioned: particle size reduction, lipid formulation, solid dispersion, and also we have salt forms optimization or selection as an add-on to the service. And finally, we can provide expert consultation at the deliver phase and also provide prototypes to be used for an animal preclinical study. Now with the assess phase, We can do various different types of API screenings. Ones that are mentioned here is that we can look at the pKa of the compound. We can select acids or base and solvents that works best with the compound. We can also look at different types of polymers that is best suited if solid dispersion is the right route to use. And also we can do high throughput screening just to see also if a certain type of polymorph is most suitable, and also if a certain type of salt form is most suitable for this compound, in improving the oral bioavailability of this compound. Here is just a little bit more information kind of detailing what can be offered at the assess phase. As mentioned before, we can do various different types of pre-formulation studies mentioned here. More specifically, which would be very important for poorly soluble compounds, is looking at the solubility at different pH ranges, and also looking at the solubility in different biorelevant media such as FASF and PESF. And also in addressing using certain types of technologies, we can look at the solubility of a compound in different types of organic solvents, and also different types of oils such as sesame seed oil for a soft gel application. And all of this assessment can be done at the assess phase using the Optiform API technologies that is offered by Catalent. As mentioned before, salt form optimization is one of the service offerings that we do offer with the Optiform Solution Suite. With salt selection, we have different types of solvents that we can test in, and different types of conditions. Okay.
So sorry.
Okay. And we have 3 to 5 different candidate salts that we can test, and we can scale up to 350 milligrams. We can do full characterization with PLM, Raman, powder X-ray diffraction, DSC, TGA, IR, and also we can do purity and salt stoichiometry using NMR, IC, LC. With one of the enhanced phases, we can also look at particle size reduction. So with particle size reduction, this is commonly used in improving the dissolution rate of the compound. Dissolution rate can be improved by increasing the surface area of the compound by reducing the particle size. So wherein the higher the surface area, the faster the dissolution rates. So the compound will dissolve much faster and get absorbed into the body much faster. However, the intrinsic solubility of the drug itself is not increased. This is where using technologies that can actually increase the solubility of the compound will be very beneficial. Here are marketed APIs that have successfully been improved in terms of the oral bioavailability just by using particle size reduction alone. I know I mentioned compounds that would fall within the DCS or BCS2A category definitely are benefited by particle size reduction. However, there are compounds that would fall in the solubility limited category, the BCS2B or the DCS2B category, wherein it could also be benefited by particle size reduction. Quite often, if using micronization alone does not help, blending the API with a surfactant, which can enhance the solubility of the API, can work in improving the oral bioavailability of that compound. And you can see cases here such as a compound very commonly known, such as fenofibrate, wherein this was very beneficial. Particle size reduction also can be used for formulation development with different types of dosage forms. You've seen examples how it can be used for oral drug delivery, as in the case of improving the oral bioavailability of a compound, wherein you would want the D90 value of the API to be less than 10 microns, preferably less than 5 microns. It can also be used for improving the dose uniformity for tableting applications, specifically direct compression. It can also be used for improving the pulmonary bioavailability of a inhalation drug, wherein you would want a very tight particle size distribution in order for the drug to get inside the lung and stay within the lung. Also, it can be used for transdermal or dermal drug delivery, wherein you want the particle size of the API to be less than 10 microns, and in order to improve the suspension and also the grittiness of the feel of the ointment or the transdermal patch. And also it can be used for ocular drug delivery, where you don't want particles very large, because you can actually see it in the patients if their particles are very large in size. So typically a lot of the formulations, they would want the API to be less than 10 microns. For an inhalation drug delivery, as mentioned before, it's very critical for the API or the particle of the drug to have a D90 value within a very tight particle size distribution. And the reason behind this is the penetration of the drug into the lung is greatly affected by particle size. So if a particle is greater, the D90 value is greater than 5 microns, it will get swallowed and it won't actually get into the lung at all. If it's less than 2 microns, and more specifically, if it's less than 1 micron, it can actually get exhaled. So it won't get into the lung at all. So quite often, when you're looking at the particle size of an API, you would want the aerodynamic diameter to be between 2 to 5 microns. So this is where working with particle size reduction, specifically micronization, can be very beneficial in improving the pulmonary bioavailability of a compound. Here are examples of different types of inhalers that have been used, and studies wherein they looked at different types of fractions of, and also how particle size can affect the performance of these different types of inhalers. With the pressurized metered dose inhaler, they saw much better performance when the particle size was less than 5 microns. With dry powder inhaler, they saw much better performance with this particular formulation when it had a high fine particle mass, or in other words, a fraction of particle size which is much more on the lower end of the spectrum, specifically less than 5 microns would be recommended. And also with nebulizers, they saw much better performance with a smaller aerosol, kind of indicating the smaller you go, the better off you are. But however, if you go too small, your API will end up getting exhaled. So it's very critical when you're doing the initial studies to know which particle size ranges, and quite often with a lot of companies that are developing formulations for inhalation drug delivery, they tend to stick to the D90 aerodynamic diameter of 2 to 5 microns. So post-particle size reduction quite often is needed, especially if you are working with an API manufacturer. Sometimes the API manufacturer does have these capabilities wherein they can do particle size reduction, but quite often a lot of customers come to us for particle size reduction because we can not only provide them mechanical milling using a Fitz mill or a pin mill, we can also do micronization. And we are one of the leading experts in micronization, and a lot of the projects that we do is GMP particle size reduction using micronization, where we can achieve D90 values between 1 to 30 microns. However, there are certain situations whereas mechanical milling is beneficial, where you would want to have a greater particle size, as mentioned before, and improving the dose uniformity. Quite often you need to make sure your particle size is pretty uniform in order to prevent segregation or sedimentation of your API from the excipients, and so you have better flowability and also dose uniformity during tableting. So Catalent Micron Technologies has manufacturing capabilities, as mentioned before, from preclinical all the way through the different phases, and finally through launch. And we also work with generic customers. Here is an example of all the different types of mills that we do have. We have the jet mill, we have the opposed jet mill, which is commonly used for inhalation drugs. We have the pin mill, we have the comb mill, we have a hammer mill. The three mentioned before are mechanical mills that we commonly use. And we also have the and the cryomill. So the cryomill is typically recommended for compounds that are pretty heat sensitive, that have a glass transition temperature or a melting temperature that is close to room temperature. So typically we would recommend using the cryomill in that application to make the API brittle enough so that it can fracture easily. Catalent Micron Technologies was founded in 1986 in UK. We globally have 15 GMP processing bays with separate cleaning rooms and temperature and humidity monitored controlled rooms. We do have nitrogen capabilities for compounds wherein you don't know the minimum ignition temperature or which could be explosive. We also have containment capabilities to work with potent compounds. We can work from 5 grams all the way to a ton, and we are GMP accredited, as mentioned here, with FDA, MHRA, PMDA, KFDA, and BISA. And we can work with DEA compounds, Schedule 2 to 5, and we also do work with cytotoxic compounds in U.S. In addition to mechanical milling and micronization, we also have analytical capabilities and offerings. We do a particle size reduction. We do crystal amorphous analysis. We can do ID testing, and we do compendial testing, stability testing. We do look at bulk powder properties. We do look at dissolution, and we do have additional services to identify the API and do any stability testing with the API. Catalent Micron Technologies has been providing this service to various companies around U.S. and Europe and for many years, and we continue to do that. And we're also always looking for new ideas from our customers and new technologies to bring in-house. One of the new technologies that we did bring in was a cryogenic micronizer to address micronization of compounds that are not very brittle and do have low melting temperatures and high glass transition temperatures. But however, we are always looking for new ideas. And also, one of the new ideas that we did have brought in from Catalent is the OptiForm Solution Suite. And this is a nice package of platform technologies that are used for improving the solubility of poorly soluble compounds. And we can provide this service wherein we can look at a new compound and address what is the best technology to use and provide a prototype for animal clinical studies, preclinical studies in 12-week timeline. So we are very excited about this new service offering, and definitely I would love to talk to you guys about it if there's an interest. So thank you so much for attending this talk, and definitely I hope you guys do have an interest in using Catalent, and hopefully the presentation that was given kind of gives a nice overview of the different types of service offerings that Catalent is offering. Definitely we do want to highlight the OptiForm Solution Suite as a good solution, particularly to small or mid-sized pharma companies who are having solubility issues with their compounds. Thank you.